Great, thanks. So in making this talk, it was my understanding that most of the people joining would be in human research and sort of interested in clinical models for focused ultrasound. So I tried to make the talk a little more broad, and I think as you go through it, it's more of an introduction to these preclinical behavioral models. Like, what are people doing with animals? What do I think is a good idea? And how can you converse with these different groups to inform your own research? So, I'm hoping you can think in your head while we're going through this, that's an interesting model. I should look for a collaboration to try that for my own research. And if you are into animal models and you're doing some of these experiments yourself, I hope that I introduce some new techniques. Maybe you have questions about how to do something. I'd be happy to answer. So this talk isn't necessarily about my own research. I do have some of my own data, but I have explored many of the techniques that are out there and trying to learn. And I've made a bunch of mistakes too. Like, while I'm trying to figure this stuff out, everybody knows it's a really new field. So hopefully my mistakes don't have to be yours if you hear about them here. I saw in the poll, at least we have 4 answers of everybody being already familiar with focused ultrasound, but I'll just go over it from a high level really quick to make sure everybody's on the same page. So what we're trying to address with this new technology are the major shortcomings of pharmacology. And illustrated in this image is that when you're taking drugs, they're bathing the entire brain. Even though you want to affect little niche circuits, you're going to get all of these off targets and all of these terrible side effects. And you can't control when they're on or off. As soon as you take the pill, you're in for many, many hours. And then there's this sort of rundown time. So I'm sure a lot of people, especially joining the BrainBox seminar, are familiar with TDCS and TMS, which are attempting to address both the temporal and spatial aspects or problems with drugs. But one of the major problems with them is that they're limited to these sort of superficial layers of the cortex for the most part. So focused ultrasound is exciting for a lot of people, including myself, because it allows us to start to reach these little deep brain areas and to hit the targets that we sort of love and hold dear. So this is really cool, but it poses a huge problem or a challenge for the field in general. And that is that now that we can access all of these different brain regions, we also have to examine them in combination with all the different ways that we can pulse ultrasound. And we have to examine them for all the different behaviors we're interested in and all of the different cell types within those brain regions. And this sort of combinatorial matrix goes on. And it's daunting. It's like, if I wanted to do that in humans, like how would I even begin to address all of these questions? And that's where choosing animal models to sort of preface the human applications becomes really important. So I've drawn up this really simplified version of the trade-offs between different model systems. And the major idea here is that when you're looking at a model, it's either very accessible or it's very translatable. Now, translatability is pretty simple. It just means if I do this in this animal model and it works, is that going to work in humans? Very basic question. Accessibility is a little more complex. That could mean something like, can I edit the genome or genes of this animal very easily? How expensive is it? Is it really cheap and fast? Or is it really time consuming and cost millions of dollars? And do they have the behavior that I want? And is it easy to record the behavior? So I'm going to briefly talk about some of our smaller little friends, which I have experience with, like the C. elegans worm, Drosophila zebrafish. But the talk sort of predominantly focuses on mouse and rats, which, if you're a human researcher, I'm guessing that's probably what you're most interested in. Some people are doing NHPs, but those can end up being just as expensive as human studies, and they're a little more difficult to work with. So again, mouse and rats, mostly because that's what I'm working at Stanford with and developing technologies around. But let's start with a little bit of examples of why on earth would I use C. elegans to do ultrasound neuromodulation research? And the power of this tool really comes in the genetics. So I don't know if anyone knows this, but you can actually feed these little worms RNA just by simply dumping solution into their little bath that they're in. And by feeding them RNA or different DNA plasmids, you can edit their genome purely through their sort of consumption of these things. So it's really easy to manipulate the genes in C. elegans. And once you've manipulated a gene, then you can sort of expose them to ultrasound. And for the most part, people look at these sort of sensory aversion movements within C. elegans. So let's say this sort of red circle here is an ultrasound field. When the animal bumps into it, does it turn around or does it go about face? And the way genetics might be important here is because you can start to knock out genes that you are wondering, if they're responsible for ultrasound sensation, or you can knock genes in that you think could lead to higher sensitivity to ultrasound. Maybe some of you saw Drosophila on the slide earlier and were wondering, how on earth are you going to get ultrasound into Drosophila? And you're not going to get it into the fly per se, not the animal that's flying around, of course, because of the reflections in air. But the Drosophila larvae sort of looks a lot like C. elegans. They're really easy to track. So here's just a video published by Kirschtein. And you can see that just motion tracking with these animals is very easy. The manipulations of genes is also really easy. There are huge driver transgenic combinatorial libraries available for fruit flies. There's a system called GAL4 where you have this driving protein. And then you'll have something called a UAS transcription element. And you can sit that in front of whatever gene and drive it in specific tissues. And Drosophila might give you a few more advantages over C. elegans because the nervous system is extremely well characterized. So you might be able to ask questions about different neural subtypes, how they're organized, and how their ultrasound sensitivity is genetically related. So we'll step even a little bit more complex here. So on the left is a video of a zebrafish swimming around. So these are, they're equally tiny. They're still very easy to track and to do simple behaviors with. But these animals are a little more complex. So they have vertebrae, and they actually have a lot of brain regions that are similar to mice and humans. I know that sounds surprising, but as an animal researcher, I think there's a lot to be translated here. And their exoskeleton is completely clear when they're in their larval stage. So on the right-hand side, you're seeing light sheet scanning microscopy. It's a pretty new technique, but that actually allows you to image the entire nervous system of this animal. And in this video on the right, the animal is actually head fixed within a little sort of bracket. And two little electrodes are implanted in the sort of dorsolateral muscles, and you can see the prediction of which way the animal wants to turn. So these animals are anesthetized. So then you can start to correlate this imaging to what the animal is trying to do. And what's also really nice about these guys is that they're in water. So if you put your ultrasound transducer in the tank, you're already directly coupled to them. And they really don't have, they don't have any bone, it's all cartilage. So there's not going to be a whole lot of reflection of ultrasound. And if you got a really high frequency ultrasound transducer, you could potentially start to target little regions within this network and to look at brain-wide responses. So I think there's a lot to be done with zebrafish. There's really not a lot out there yet, but I've heard a lot of discussions about all the great things that can be done. Okay, so let's start in with mice. Mice are very easy to track behaviors, and here's a really simple one. On the left-hand side, there's the shavings that they're used to, and on the right-hand side, there's a sheet of plexiglass. So they really don't like being on plexiglass. It makes them kind of scared and is aversive. But you might imagine if you're delivering ultrasound when they're on the plexiglass side of the chamber, which you can sort of imagine is on when this light is flashing, you can then ask, do these animals start to bias their position towards the plexiglass? So that's sort of a question of, do they like the ultrasound stimulation? Is it rewarding? And you can do the flip experiment and make it aversive as well. So I'm showing you guys tracking because it's easy to just visualize and feel out a sense of behavior in animal models through tracking. But there are many well-characterized behavioral paradigms for mice. there's things like food intake, addiction, fear, memory, aggression. It goes on, and I didn't go through how to do all these today, but I do have experience with most of them if any of you have questions later. So it always becomes the question of, what am I most interested in, and where is that brain region, and how do I measure that in the context of ultrasound? And when you're trying to design these experiments, I would advise you just to look at previously published experiments with dreads. So these are basically designer drugs where you genetically encode a receptor for the drug within a spatial subtype of the brain. So it's kind of like ultrasound. It has this spatial aspect, and it sort of has a long time scale. Similarly, optogenetics can be targeted to little brain regions, or even bipolar stimulation, like these little electrical fields. So for ultrasound, if you don't know where to start, look at your fields, find one of these experiments and just copy it, and just target your ultrasound to that brain region. And we'll get into how to do that in a little bit, but that's sort of a preface on that methodology. And then, of course, I wouldn't advise thinking about experiments as an all-in-one. You don't have to measure your neural response while you're looking at behavior. You could do the neural response and then try to correlate that to behavior afterwards, which is pretty common. And a big red flag advisory here. So I actually will credit this idea to Mikhail Shapiro, who we talked about this in a lecture recently that I was a part of. Ultrasound has mechanical effects. There's no doubt about it. It's really, really difficult to mask them. I would argue that you can never fully mask them. So if you've ever, like held an ultrasound transducer to your head or to your hand, even if you don't hear it, you can feel it. So there's this sensory component that is outside of audition. And whatever it is that we're doing, with our ultrasound, we're trying to do direct neuromodulation. So it's shown on the left here. But we're definitely going to get a little bit of overlap in this auditory sensory region. So I think I want you to remember that when you're thinking about experiments or looking at someone else's, that whatever the phenotype is, hopefully it's not related to these two on the far right. So try to avoid those behaviors that are sensitive to, an animal being startled by sound or feel. So I'll jump into a little bit about best sort of practices. Ultrasound transducer elements can be bulky, and they require very high spatial precision. So you're trying to target these small fields in the animal brain. So I often talk about how to use head fixed animals for a starting point. So head fixed, and I'll show you a little bit about how it's done, but that's really when you're cementing, using dental cement to fix the animal's skull in place. And that allows you to use immobile equipment, such as imaging or sensitive electronics. It allows you to also keep your brain coordinate system fixed in space. And often in these setups, the animals can still perform some awake behaviors, although they are constrained. So really briefly, what is stereotactic brain positioning in mouse? I bet everybody already knows, so I'll be really brief. The one advantage of the mouse is that the brain is extremely well characterized, and it's hardly ever different. So it's not like a human where some people have really big skulls and really different brains and some very small. All of these mice look pretty much the same. So if you know where you are on the skull, you can basically point your beam and have pretty good confidence that you're hitting, some region that you're trying to target. And of course, to do that in animal models, particularly the mice, we usually head fix the animal and open up the skull, and we'll look for these two fissures on the skull called bregma and lambda. And relative to those points, we have this sort of map. And if you put in an unequivocal plane across these 2 points, then you can mount a sort of bracket to the skull. And that bracket will allow you to not only head fix the animal by putting little screws through this metal bracket, but the bracket can also contain sort of a mount for your ultrasound equipment. So examples of that here. So here's a non-head fixed animal, and I don't have a video of this, but this animal's got a lot of stuff on his head and he's not super comfortable. And every time these bipolar implants that I have here bump into the side of this cage, I would get like huge artifacts. even if I electrically shielded it, it was still a problem. And here's my ultrasound cable as well. And you can see that I have these little head bars, and I have an ultrasound sort of bracket on the animal's head. So it becomes a lot easier to work with, much lower noise when you've head fixed the animal. So here's an example of a system that you can just sort of buy off the shelf. You screw the animal's head bracket in here. And in this image, I was just floating a styrofoam ball underneath the animal. And that basically means you have the styrofoam ball, you're shooting air underneath it, and it's creating like this sort of rolling surface for the animal. And you can still track motion by looking at the ball and how dots that are visually represented move. Even simpler than that, you can create these like treadmills with sort of off-the-shelf parts from Thor Labs. And here we're just looking at like a ribbon cable, and the animal can pull on that cable and walk comfortably. This one's much easier to do. It doesn't require all these fancy, ball things. And for me, both of these have worked very well. I can still do freely behaving, but the more stuff you get involved, especially if your transducer is very large, the more you want to lean to these two right-hand scenarios. And if you want to get really fancy, there are almost off-the-shelf systems for VR for mice. So here's an animal on a ball, a paper published by Guifan Chen, where the animal's running on the ball and you're tracking its motion. It's head-mounted to a confocal imaging rig, and at the same time, a projector is sort of shining this arena in front of the animal. And to start, depending on however the ball moves, you can actually orient the screen. So the animal thinks it's running through virtual reality. And people are using this for decision-making paradigms, spatial tasks, things like that, if you want to get really fancy. Another really fancy tool developed by Jacob Voits from Mark Carnett's lab is this head-mounted bracket that actually senses rotational strain and uses a servo to actually adjust the position of the head mount. And the animal's running on a hockey puck floated on an air table, so it's really easy for him to walk around. And at the same time, you can image whole cortical regions, with this device. So I know it's rotating, but you can easily correct that. And the image on the right, so I'll just back it up a little, it shows what that correction looks like. So now you can actually look at specific regions within this brain. So those last two are much more complicated, but there's just some options for you there. Okay, so what about different neural activity readouts? What are people working with in the field, and how do they compare to each other? I'll just start with the very typical electrophysiological methods and why I wouldn't recommend them. The principle between electrophysiology methods is just that the cell acts as a resistor, and there is a voltage across the membrane. So you can measure that voltage difference within an individual cell, or you can try to measure those little voltage differences across broad fields of many, many cells, where the aggregate field potentials sort of add up and summate. And most people, when they hear electrophysiology, they might be thinking sharp electrode physiology. That's where you take this little sharp glass electrode and you poke it inside the cell. And then you have another reference electrode that you're putting in the bath solution outside of this cell. And you're basically measuring what this individual cell is doing. And here you can see they'll actually inject a current into the cell. You'll get this stimulation artifact. And you can measure the amplitude of that cell that you're poking there. And another, even more finely precise measurement of membrane potential. So rather than this bulk difference, you can actually measure the voltage at the surface of the membrane, is to sort of suck onto the membrane with a glass pipette and pull out the piece of membrane. You're almost becoming part of the membrane solution, and then you can measure relative to the outside, and that's called patch clamp. So patch clamp you can do. Merit Madouk from Stanford does a lot of patch clamp with ultrasound, where she'll have an ultrasound come in from the side and blast the whole cell with it. The caveat here is that you can't use low frequencies because the glass pipette will resonate with anything in like the sub-megahertz range. And she typically uses 43 megahertz, which allows this to stay in place because the glass doesn't vibrate. but that's not something that human researchers are using. It's a really high frequency, and you begin to wonder how relevant it is for the human system. The one on the left here, the sharp electrode recording, is probably more appropriate, but there are drawbacks. So there was this study by Morgan Collins where he basically filleted open a leech. So here's a sort of prep where he's looking at these things called Retsea cells. They're like huge neurons within the leech. And what he's doing is putting it on this prep, lowering in a glass electrode to do these sharp electrode recordings. And he has a transducer coming from the bottom here, a focus transducer, creating a field over the leech while he records these sharp electrode changes. And the ultrasonic field looks something like this. Again, here's the leech and the recording electrode. And the problem is, here, with this pink line here, you can see a very short pulse of ultrasound. You can see that indeed the cell does respond with a deflection in electrical potential. But unfortunately, he was able to do very similar things by simply moving the pipette up and down. And the reason that this is a problem is that if you're hitting this thing with ultrasound, you're going to get some radiation force upwards, and you're actually going to move the membrane around the glass electrode. So you're going to have an effect that looks a lot like just moving the pipette up and down. And now the question becomes, is this because of neuromodulation, or is it because the pipette is simply moving relative to the prep? And here you can see, he very well mimicked what he saw in terms of depolarization by increasing ultrasound pressure with simply displacing the pipette, a number of microns. I would argue that neuromodulation is probably still very possible in this PRET, and that he had to use very low pressure, 60 kilopascals, 80 kilopascals. I would really not expect much of a neuromodulatory response at that pressure, but it is nice that they highlighted these artifacts that you can get in that low range. So if you are doing electrophysiology, and I've done a bunch of it, I would really recommend looking at local field potentials. So that's different in that you're just sticking electrodes in this bulk mass of many, many cells. Here you can see a tetrode from a Busaki paper from 2004. And you're not actually puncturing any cells. You're sort of measuring the net field of a local area shown in pink here relative to some ground electrode that's sort of off-site here. And it's really ideal if your electrode isn't underneath your ultrasound field, because you can still imagine that if ultrasound was pushing on this tetrode, it's going to move this around into different cell clusters, and you may actually get some artifactual response. Alternatively, if you only look at offline effects, so if you look at what happens after the ultrasound is delivered, then you're probably looking at something that's real. Although it still stands to reason that maybe you're looking at something that happened because this electrode moved and pushed the cells around. So, it's kind of like the whole auditory thing. There's probably some hybrid if you're using electrodes of, okay, the thing's moving a little bit and the cells are moving, and also there's probably neuromodulation. But it's something to consider that, it's in there, it's out there. And my experience with this has been mostly in epilepsy. So I have done a bunch of different epileptic models where I'll either use electrical or kainate acid basically to damage in real time or chronically cells. And for my kainate acid experiments, I'm basically looking at local field potentials in the hippocampus. And here you can see these sort of large oscillatory events that have these riding high frequency oscillations on top of them. And these are very typical seizure events in the kainate acid model. And looking over time, I can deliver, let's say, like a 1 hertz stimulation with ultrasound for 5 seconds. And here you can see basically a bunch of epileptic events stacked up over many, many trials, and whether they occur before or after the event. And you can see that the ultrasound basically shuts them down hard. The caveat here that I'm thinking about very often is whether or not this startle or this sound that they may be hearing stop seizures. And I'll tell you up front that it definitely slows them down. And most of my findings have to be a comparison of, does ultrasound do things beyond an animal hearing a sound? So aside from all the issues you might experience with electrophysiology, you may be thinking, well, what if I can do imaging instead, which obviously would have way less of the issue based that electrophysiology would have. So I'll try to introduce imaging to you guys, especially the human researchers who may not have a whole lot of experience. And the principle here is that if I have a fluorophore molecule genetically encoded in, let's say, neurons of interest, or let's say even the whole brain, I can then shine in an excitation wavelength and basically raise the ground state of these molecules into an excited state. And when these molecules relax, they emit back basically a longer, lower energy wavelength. And the excitation emission spectra, might typically look something like this. So here's GCAMP. This is genetically encoded calmodulin. Well, I forget what GCAMP actually stands for. I don't know if it really stands for anything, but it's a very typical fluorophore used. And what's interesting about GCAMP is that it has this binding domain for calcium that changes its configuration when calcium is present. So it basically only really receives light and emits green back if calcium binds in the pocket. And calcium being present inside the cell is a strong indication that that cell is active. So this is an optical readout for neural activity proxy. And a very typical calcium, fluorescent response would look something like this. There are many versions of GCAMP. So there's GCAMP slow, GCAMP fast. And you can see that for GCAMP slow, it aggregates calcium much slower and the signal will last longer. GCAMP fast is better for like fast spiking neurons. It's a much quicker response, kind of flips on and off very rapidly. And working with this data, what researchers will typically do, including myself, is to have some baseline period and then to have a stimulation event. In our case, that would be ultrasound. And then to look at the aggregate or area under the curve following the stimulation, and then just to compare that to baseline. And you can use this equation down here if you're interested in doing that. And you're never really just looking at like one cell where you get this really clean response. You're usually looking at a bunch of cells that summate in space and time. So your aggregate signal will look much noisier than this shown on the left. And this is like, I forget, like 10, 11 years ago. This is Jamie Tyler showing that if you cut out slices of the CA1 and have calcium indicators, and you can see when he's sort of turning on the ultrasound, you can get these little flashes of light in these individual cells. And this is sort of an idea of what it looks like in response to ultrasound. And it's really easy to work with this data, especially if you have images rather than bulk optical collection. Just to give you guys an idea of how to work with it, you'll basically look at a stack of individual images, and then within those images, you can draw these little regions of interest. To show you guys how easy it is, I actually downloaded Jamie's video from his paper and loaded it up into ImageJ. It's free image analysis software. And I drew my own little ROIs and quantified that fluorescence change over time. And here you can see on the right, you can almost tell exactly when the ultrasound went on. So it went on right here. You can see that the cells sort of all rapidly respond, and the cells are circled over here. So it's really that easy if you have a picture of the brain. The idea being, I highly recommend, if you're interested in animal work, moving towards calcium imaging and having your ultrasound come in concert with that imaging. A little bit more about how to work with it specifically with ultrasound. Here's one of my own calcium traces from an experiment. You can see that it declines over time. So that's actually photo bleaching. So these bonds within the GCAMP molecules will spontaneously sort of flip out or break. So you're always, when you're thinking about these experiments, you're always doing a trade-off between how much excitation light you're passing in, and your signal to noise ratio, and how that affects photo bleaching and this downward trend. But the principle I've been using for my work, and it works really well, is to have series of ultrasound stimulation events, let's say. And I don't know if you guys can see my mouse. I hope you can, because I've been using it a lot. But if you can imagine little ultrasound events happening and then stacking up all of those events relative to your ultrasound stimulus. So here is me looking at the thalamus in response to ultrasound, which is this gray bar here. And I have 20 events to look at. They're all super consistent, and like you can see that they work. And I'm actually taking the baseline and just normalizing it out to this sort of pre-period. And you can then take an average of that over time. So that will tell you many things. You know, one of them is, how long does it take for ultrasound to sort of aggregate a response and excite neural tissue? You know, does that last after the ultrasound pulse, or does it stop right away? And then you can go in and start to ask questions of like, how much intensity do I need? What do different pulsing schemes look like? All of this stuff I found works really nice with calcium imaging. You know, as compared to electrophysiological approaches, which we can talk about later if you guys have questions. I haven't seen any work with genetically encoded voltage indicators, but I wanted to let everyone know that they're out there and they're getting much better. And the idea behind these voltage indicators is that if the membrane is highly hyperpolarized, I'm assuming everybody has a basic background in neuroscience, this means the cell is very unexcitable, I guess you could say. There are rotational shifts in some of the bonds of these GEVI indicators, and that actually changes how much they will fluoresce in response to an excitation. So here you can see the flicker 1 genetically encoded voltage indicator, and you can see over time when this neuron is spiking that you get this really high resolution millivoltage change. On the bottom, they're actually doing electrical, they're doing a patch recording while they image this indicator. And you can see that even optically, it's highly faithful to what's happening electrically. And I think someday these will probably replace things like GCamp, because they're such high resolution and they're really good. But right now, the signal to noise ratio it's like absolutely garbage. I mean, comparatively speaking, someone would probably kill me for saying that who's really into these things, but they're really difficult to pick up. And again, they have the advantage though of being really sensitive to membrane potential. So that's kind of the upside. An important distinction between the calcium indicators and the voltage indicators is whether or not they're fast enough to pick up single spikes. It sort of depends on the field that you're looking at. And if you're using GCamp fast, maybe you can do that with calcium indicators, maybe not. With the GEVIs, you definitely can. So I hope there's a researcher out there thinking about using these. I'd love to see it someday. So I'm going to put together a bunch of these concepts in an experiment by one of my collaborators, Jordan Farrell. So here he has the animal head mounted. He's doing confocal imaging of the animal's brain, and this is an epileptic animal. Here you can see him actually having a seizure. He's got these mouth twitches and whisker twitches. And he's also recording the speed at which the animal runs on this ball while doing a local field potential. So I know you might be saying, God, that's a lot of stuff. But imagine imaging individual cells, animal motion, and local field potentials while delivering ultrasound. Like, that's going to be a good paper no matter what you do. So I think I would highly encourage people to think of ways to integrate all these systems together in experiments as elegant as this. And I know I haven't really talked a lot about how you get ultrasound in, so I'll just point to it here. But you can easily imagine passing a ring around this confocal lens. And I don't know if everyone knows this, but a ring, depending on the internal diameter, will create a natural focus in space. So something I'm doing and other people are working on is actually putting rings around these confocal or imaging modalities and then focusing sort of deep in the brain while performing all of these cool recordings at once. It's really an attractive idea. The other reason I really have been attracted to imaging is because you can do dual imaging of different cell types. So many people would argue that electrophysiologically, you can still differentiate cell types. It becomes much harder when the cells are similar. So you can imagine, like, what if I'm trying to distinguish D1 versus D2 receptors, or expressing neurons in a similar field? The size is similar, but so you might not see that electrophysiological. Here, I'm doing dopaminergic versus GABAergic cells at the same time. So I'm flipping two different fluorophores on and off really quickly. And here you can see that when I deliver the ultrasound, this one cell type responds really strongly, very immediately. These are GABAergic neurons. And they may actually suppress the activity of these dopaminergic neurons because this response doesn't start to happen until after the ultrasound is off. So I'll just give you guys the forewarning. There are really, really crazy, interesting interactions between cell types all across the brain. It will take us many, many years to explore all of them, but I think it's a wave that is coming. And it's particularly important for human researchers because any brain region you look at, in the past, you might have been like, I'm doing TMS, I'm going to hit all the cells. But with ultrasound, you can see here that they're not always going to do the same thing with different cell types. And that may give you the ability to sort of get cell type selectivity that you may not have otherwise had. This is a really expensive option. I'm just going to finish up the different measurement methodologies by saying that you can definitely do PET scans. So I've done PET scan experiments in the animal brain with my setup. So, a typical experiment might be like you're injecting fluorodeoxyglucose, a radio-labeled glucose molecule, into the animal, and that as it's sort of collected and bound within the brain, you now have this sort of radio emitting molecule within that region. So if there's a really active region in the brain and it's got these little molecules, you can do that association. So you might deliver your ultrasound stimulation during behavior or what have you, and then you pop the animal onto this little board, send them into the PET scanner, and that will spatially localize where your radio emissions are coming from. I'm not going to go into how these work, but if anyone has questions on that, I can tell you about PET scans. They're, in my opinion, I don't know if like Leonard Verhagen does really great work with fMRI. He would kill me for saying this, but PET is really like much higher resolution and probably a better indicator of brain activity relative to something like fMRI, where you're just trying to measure blood flow. And here's just a sample sort of brain from my own work. Here I was targeting the hippocampus with ultrasound, and I was working with Juan Gomez and Mike Michaelides over at NIDA, and you can see that you're getting much less uptake in the region where I'm stimulating and probably a little bit below in sort of the thalamus here. And you might look at this and say, oh, okay, yeah, I can see it. It's not that great. But if you've done optogenetic experiments or dread experiments and you've done PET scanning, you would be shocked at how good ultrasound works for revealing these patterns. And a way to statistically work this out that's sort of fun is you can actually just rotate this brain along the z-axis, like flip it over and land it on top of itself, and then just do a statistical voxel-wise comparison to say, okay, what's the difference between these two hemispheres? And that's the approach we've been taking. I'm going to spend very little time on this because I don't love this technique. You can also do c-Fos in animal models, and you can't do that in humans because it requires you to kill the animal or sack the animal and pull the brain out. And what c-Fos is it's an immediate early gene that is expressed. So when a neuron is active, it starts to express this gene over a period of 90 to 120 minutes. And then you can, again, you can pull the brain out and just use antibodies to look for it. I'm sure many of you are familiar with it. The annoying thing is, like, it's a lot of work to pull this tissue out and to do this. And the reason people are still doing it is because it's sort of cheap and you don't need imaging equipment or fancy stuff to get it to work. But it has really bad temporal resolution. So you're like aggregating this post effect over 90 to 120 minutes. And a lot of people don't know this, but c-Fos is not equally expressed across cell types. So if you're like doing c-Fos and looking for GABAergic activity, I don't think that's going to work very well compared to like glutamatergic activity. I hope people move away from this, but if you're still doing it, this is an experiment from a collaborator of mine, Kim Butts-Pauley, where she's stimulating this region of the brain in control animals or compared to sham control animals. And you can see here that you're getting these little dots. This is like c-Fos activity in this brain region, only when there's sonicated tissue. So, it works, but I wouldn't recommend it. One of the reasons I wouldn't recommend it is because in a lot of my work, depending on what cell types you're looking at, so here on the right is GCAM imaging from a set of cells in the thalamus, and the ultrasound bar is, the ultrasound comes on the gray bar here. You can see that you get this fast excitation in these neurons. So you're getting an increase in activity that normally you'd say, oh, okay, then I should get c-Fos, right? Like they are responding. But after I stimulate, you can see this refractory drop in neural activity. And at least for this experiment, it goes on for quite a little bit. Like it's well beyond 10 seconds. So if I summed up what's happening over this window, I may get a net zero excitation. I may get more, I may get less. It's really a little bit of a black box. But because c-Fos is an aggregate over this window, I may miss entirely what's happening. And what's happening is, I'm sure to everybody looking is really exciting stuff. You're getting this fast response and this inhibition. So you can't see that with c-Fos. I recommend people move away from that. Okay. This is a little bit less for our human researchers, so I'll try to be quick about it. But for the animal researchers, how do I think about ultrasound hardware and what do I need to get started? If you're in the human sector and you have access to an animal lab, maybe this will be helpful for you to think about how much you have to set up to get this going. And it's really not a whole lot. You can set this up on a pretty low budget. For my own work, the majority of the equipment is shown here. It's like you can have a laptop, 2 function generators, which I'll show you some examples of. In many cases, you may want 2 because you can basically modulate a continuous frequency. Let's say I have 500 kilohertz here with an envelope frequency. It's where you would multiply these 2 together and you'd get this smooth waveform. And I know I didn't get into this today. It's a little bit out of the scope of the talk, but smoothing the edge of these waveforms basically stops the animal from hearing these continuous wave pulses. So we think. And you're feeding this envelope into an amplifier. This is going to be the most expensive equipment. And then you're basically passing it onto your ultrasound element or transducer. You can put a matching network in between. So I do. This is basically to reduce electrical reflections from the electrical impedance mismatch, because ultrasound elements are like supercapacitors. They look very different from wire, and electronic reflections will happen at that interface, but you can block that with a matching network. So people, like when I'm collaborating with people, they'll ask, all right, well, what do I need to buy? I always recommend really like these basic things. The Agilent 33210A, it's a phenomenal little function generator, and it has this $100 interface that's USB compatible, where you can plug that into your computer and use Python to control everything that this does. So for me, I've always used that in a way to basically control experiments. So I just hit play, and then the thing will, it may change the features of ultrasound. It may increment up intensity or change pulsing frequencies, stuff like that. It's really good to work with. I'd be happy to share the software for controlling this that I've written as well. Power amplifiers, brand new. These things are pretty expensive. I think brand new, like this particular ENI 2100L. This is a kind of amplifier that will give you the power you need without knowing anything about your ultrasound transducer. This can cost like 20K brand new. I've bought old ones from like the 70s that work fine. These things were built to last. Those cost like 2K, a little cheaper. And then transducers, like, oh man, there's really a lot of options for you out there. I actually think I have a shot here. So when I was looking at different transducers, there's a company called STEM Inc. They make these sort of curved array, or not, I guess, curved single element transducers. These are like little piezos that have a natural focus. at a given depth that you could look at. I mentioned rings having a natural focus relative to the internal diameter. Tons of companies making these rings. With both of these, you would basically attach a positive and a negative to either side of this element and then just oscillate the voltage, and these will expand and contract and emit ultrasonic pressure. Olympus makes like a little bit more plug and play transducers. They're immersion transducers. They were really made to detect flaws in material surfaces, I think predominantly plumbing actually. Nevertheless, you can repurpose the focused ultrasound transducers to sort of angle them in to the brain, and I can point you guys towards those if you're interested. Sonic Concepts, who's actually partnered with BrainBox for the NeuroFus, they make a bunch of really little transducers if you're trying to make something for mice and rats. And as many of you know, this scales all the way up to use in human researchers. I'm sure some of you listening today are already using these devices, and they're really spectacular, from what I understand. I thought I'd include this because this paper was published like a year ago. It's a really fancy little method that I saw from Ying Zhang. I think he's from somewhere in Boston. I think it's like Boston College or something. Anyway, the technique they had is sort of using these optoacoustic principles where he shoots a laser through a fiber optic cable, and he coats the end of this fiber optic with 2 layers of like an epoxy laced with graphite. And essentially, when you pulse it with a laser, you heat up these layers differentially, and basically this fast expansion contraction creates displacement. So like if you can imagine this thing blowing up a little bit and then displacing the water around it, and you shut the layer off, it quickly shrinks back. And by pulsing this really quickly, I believe in his publication, he pulses it at like 5 kilohertz. You create these actually complex ultrasonic frequency patterns. So here you can see the magnitude in decibels. So you sort of have a logarithmic scale here. And you can see that the predominant frequency produced with this tool is around 1 megahertz, which I would argue is very relevant to many of the tools that human and animal researchers alike are measuring. And here's, forget what they call these diagrams, but basically this shows you relative to the front of the epoxy, which is at 0 here. The red basically shows how much we're getting out of the front versus in reverse, which would be 180. So most of the pressure is being pushed forward, but you get this omnidirectional sort of field. And the idea would be that if you put this really, really little probe, you can see the scale here is 1 millimeter, it's very small, like directly next to your neurons, or implanted this fiber somewhere in the brain, then you can do local stimulation of these cells. And I'm really excited to see this implemented in freely behaving animals. So in this paper, it's only done in anesthetized fixed animals. Here you can see sort of the ultrasound pressure wave. I guess I just repeated this. I was going to talk more about it. But anyway, that's a really neat technique. There's a bunch others. I only said a few here just to give you an idea of what people are using. I'd be happy to talk about others. And then for coupling to the animal's brain, I think if you're new to the field, this is a little bit daunting. And I would advise you to not be scared about it. There's like a ton of ways to couple to the skull. You can, the way that I do it is really just that I 3D print these little brackets and I fill them with gel or Vaseline or whatever. You can't use water in most cases because it'll bleed out. But you can just throw gel in there and then stick your transducer in. If you have a really big transducer, so this is from Kim's lab, showing you one of her preps, you can do something like creating a little water sac and filling this with degassed water, and then just sort of smushing this against the head, and then put some ultrasound gel in there. But again, I would just say that you don't have to be scared about how good the coupling is. Like at first, like I would experiment with stuff and just try to measure whether or not you're getting ultrasonic coupling using like a hydrophone and like an extracted skull. But be creative. There's a million ways to do this. Obviously, it's going to change on what experiment you're doing. Okay, so I think that's it. I don't know how I did on time. I had no clock here. I hope I didn't go way over. There's more resources available through iTrust, which is partnered with BrainBox. And Kim and I have a course that sort of covers a lot of these topics. It's on YouTube. If you just Google ultrasound neuromodulation, you'll probably find it. And a huge thanks to Dan Reese and Andrew, who I didn't get to catch up with, but Andrew had originally asked me to the seminar. And I'm really glad he did. It was a lot of fun. And thanks so much. Here's my email if you have any questions. So I'm gonna stop screen share. Dan, thanks so much. I don't know if you want to read me questions or how you want to go about this.
Yeah, no problem. Thanks so very much. Thanks again, Keith, for that. That was great. I will just take back the presenter role. There are a couple of questions that came in during the talk. Again, feel free, everybody, to pop in any extra questions that you might have. I will look for the first one, Marco, who I just lost from the list. Maybe he just got disconnected. So okay, I'll go ahead and answer that question for him. So he just asked quickly, have you used fiber photometry to monitor ultrasound effects?
Yes, I have. That's one of the main approaches I'm taking. It works awesome. I know there's a bunch of labs doing it. Yeah, I have. I don't know if I have too much more to say about it, unless Marco is still here. If you have specific questions, it works fantastically well.
Cool, thank you. If he pops back up, I will give him a nudge. And then Moises, I will try and allow you to use your microphone. You can ask a question directly. It normally takes a second for the system to wake up.
I think I can actually see it over here myself.
Yeah, no computer audio. Okay, so so really like the talk, which is nice, and I think it seems like this technique is quite new to him. Just I think you did touch on it a little bit later on in later slides about use of ultrasound in human research. Do you have any direct experience of that? I know that there is from the BrainBox side of things. We know that there are publications and so on with using ultrasound for human work. But anything that you can comment on from your experience, Keith, and kind of where you might want to point, what direction you might want to point him in as well.
Sure. So I'll speak just a moment from experience. So like I think you weren't asking this, but have I personally experienced it? So I do have some friends that have like personally tried it. I think Giselle is actually on here. So she's actually held a transducer up to her own head and given it a go. I've put my hand in like tanks, like old school ultrasound researchers used to do. And I mean, like, I don't know if I can legally recommend this, but it's bizarre. It's awesome. That's a good way to like experience ultrasound neuromodulation in peripheral nerves for yourself. I think the question was actually about human research, which is ongoing and really growing rapidly. Honestly, I think BrainBox is going to have a huge part to play in that. So they're designing, or they've built an IDE, so an investigational device exemption human device that can use neuronavigation, and you can hold it up to your skull and kind of have an idea of where you're targeting. The reason I think human research has been slow up till now is because people are underpowering the ultrasound big time. Not because of the FDA. The FDA or the European regulatory agencies actually allow quite a bit more than human researchers are using. And that's why animal models are cool. I don't know if this is ethical to say, but we can go right up to these limits and nobody bats an eye. And let me tell you, the effects, they're much better as you go higher up. So the other part is, and Pablo Villaslata, one of my co-founders, is here. We have started a company to try to translate some of our findings in animal models into human devices. We're really interested in what BrainBox has developed as a preclinical tool, or even a clinical tool to assess how much these things will work. And I think there's a lot of other tools from other companies, including like Brain Sonics and Fraunhofer, that are being made for human work. I'd be happy to send you what human papers are out there so far, but there are more coming. That might be the last of the questions.
Yeah. Thanks, Keith. That was really great. Overview of like the human work, and good to have your input on that. Yeah. So if there are any more questions, yeah, please feel free to just drop them into the chat there. There's one more poll question, maybe some of you have answered already, about which hopefully now you can see on the screen, how likely to incorporate TOS into your research in the future. Yeah, if you haven't answered it, then you should be able to do so now. Some people are already underway with it, as we know from some of the attendees on the list here, and yeah, maybe a few waiting for clinical indications as well. So I'll just show those results so everybody can see kind of the spread. I think just from this 1 hour, we know that there's a lot that's still to be learned, and a lot of research still to come and ongoing. So yeah, it's definitely an exciting field. We're very glad to be involved with it on the brain rock side as well.